Hi, I'm Dr. Brian Kaufman, a retired family doctor and a longtime CLL patient myself, and the co-founder, chief medical officer, and executive vice president of the nonprofit CLL Society. And today I'm going to talk to you about some interesting and important papers from the American Society of Clinical Oncology's annual meeting in 2022. The next paper I'm going to discuss is Preclinical. Its title is Preclinical Activity of the Irreversible Menin Inhibitor BMF-219 in Chronic Lymphocytic Leukemia. So we're going to start in this patient-friendly summary with the bottom line. BMF-219 is an irreversible, that means it binds in a covalent way irreversibly, and it's an irreversible menin inhibitor, and it demonstrated strong preclinical activity against CLL patient specimens, including those with high-risk markers. Who did the research and where was it presented? Priyanka Saumananath. She's a PhD who works at Biomea Fusion. They're the manufacturer of this chemical, and her colleagues presented the results at the American Society of Clinical Oncology, ASCO's annual meeting in Chicago in 2022. What's the background? For many of you, this may be the first you're hearing of menin, but it may not be the last. Menin is a widely expressed protein found mostly in the cell's nucleus that has many activities, but one of the things that it does is downregulate the expression of BCL2. BCL2 is overexpressed in CLL and blocks programmed cell death or apoptosis. Blocking BCL2 is how venetoclax works. What were their methods and who participated? There was a comprehensive panel of CLL samples that were isolated, taken from patients with early to late stage disease, including those with relapsed and refractory disease. They were cultured outside of the body, ex vivo, in the presence of BMF-219 and in the presence of other reversible menin inhibitors to assess the anti-leukemic activities of these compounds. And what were the results? BMF-219 demonstrated very high potency, achieving a 98% cell lethality at low exposures in all the patient samples that were tested. These were exposures that could be reached inside of patients. Specimens isolated with patients with clinical profiles of high-risk genetic backgrounds that are generally associated with inferior outcomes to standard therapies, such as mutations in TP53 and Notch1, and chromosomal abnormations such as deletion 13q, trisomy 12, and complex karyotype, exhibited high sensitivity to BMF-219. BMF-219 was also highly effective in those patient samples from those who were resistant to bendamustine, ibrutinib, or venetoclax. So what are the conclusions and comments? BMF-219 is an irreversible menin inhibitor that shows promising preclinical data suggesting it might become an important novel therapeutic option for patients with CLL. For comparison, all the specimens that were also exposed to different reversible menin inhibitors did not have nearly as strong results. Still, CLL cells are notoriously easy to kill in petri dishes and much harder to kill and eliminate in the patient's blood, marrow, and lymph nodes. Fortunately, clinical trials for CLL are in development, and the CLL Society welcomes research with an entirely novel mechanism to eliminate the cancer, because despite all the progress, CLL is not a solved problem. In the way of links and resources, we give a link to the actual abstract, which you can find on the website by clicking on its title, Preclinical Activity of Irreversible Menin Inhibitor BMF-219 in Chronic Lymphocytic Leukemia. Thanks for listening. Stay strong. We are all in this together.